He's a professor in molecular and cellular pharmacology at the University of Oxford and also a tutorial fellow in medicine at Worcester College, Oxford. So he has been publishing so many peer-reviewed articles, and also he's quite into population science and communication, the science to the public, and with this quite deep knowledge in genomics, proteomics, embryogenesis, and also in population science. We're very pleased to welcome you to today's lecture here in University of Tartu. And today's topic is about TPCs, endolysosomal calcium channels that play key roles in health and disease. So over to you, Dr. John.
Thank you very much. Thank you for the invitation to speak. It's great to be in this lovely city. So what I'm going to talk about today is a family of proteins that we've been studying. Why is it not working? It's not viewed. That's why. A family of proteins that we've been studying for some years now. But what really led us into this interest in this particular family of proteins called 2-pore channels, and I'll explain a bit more about them soon, is that I've had a long-standing interest in the role of calcium as a ubiquitous cellular messenger in actually mediating all sorts of different processes. So these are just some of the areas of research that I've been studying over the last few years. Actually, like Ali raised there, my original interest was really only in reproductive biology. I was one of the people who identified PLC-Z2 as the factor in the sperm that activates an egg to develop into an embryo. But because PLC-Z2 is a calcium signaling protein, that led to a more general interest in calcium signaling. And so, as you can see, there's a whole number of processes as shown here that calcium signals regulate within the body. What I'm going to talk about today, and I should say that all of these different things we found that 2-pore channels are involved in. What I'm going to talk about today is, first of all, some of my recent work on cancer, which is quite a new area for us, and then a bit about the role of the same set of proteins, but in reproduction. So just to give you a background about what we mean by intracellular calcium signaling, these are just some of the key messengers that play roles in the cell by releasing calcium from stores within the cell. So it's been known for some years that the endoplasmic reticulum is an important calcium store. And the first calcium mobilizing messenger to be identified was IP3 back in the 1980s by Michael Burridge. And it was then shown that phospholipase Cs are the enzymes that, by breaking down the substrate PIP2, can generate IP3. And PLC-Z2, which I mentioned already, which is this factor in the sperm that activates the egg to develop into an embryo, is a form of PLC. So that's how this works. Subsequent to that, it was discovered there was another main calcium channel on the endoplasmic called the ryanodine receptor. And work by my colleague Anthony Gaglione showed that this is activated by a substance called cyclic ADP ribose. Now, another calcium mobilizing messenger that Anthony Gaglione started to study is called NAADP. And with him, we showed that its target within the cell is the 2-pore channels. Now, the interesting thing about the 2-pore channels, these are calcium channels, is that unlike these which are on the endoplasmic reticulum, TPCs are on the lysosome. And that has led to a more general interest in our group in endolysosomal organelles and their role in cell signaling. So this is endolysosomal trafficking. Another big interest in this department in endoplasmic vesicles, endocytic vesicles. And obviously there's a link here because EVs are released from cells as part of the trafficking system, but we've got a particular interest in the ways that these endosomes and lysosomes play a role within cell trafficking, but also because they're also a calcium store, and they play important roles in cell signaling. And in particular, we discovered when we were looking for the target receptor for NADP that this appeared to be a novel set of proteins called 2-pore channels, TPCs. So these are interesting in that they are, well, it's a family of just 2 of these proteins in humans. and other mammals. And what we've found is that they have different locations in the endoplasmic compartment. So whereas TPC2 is mainly concentrated in lysosomes, TPC1 is found in recycling and early endosomes. And this may, of course, have implications for their function within the cell. And what we're starting to find in all sorts of different systems is that TPC1 and TPC2 may play quite differential roles in a whole number of processes. So they're not just, substituting one for the other. They seem to really play quite different roles in the cell. And this is linked, we think, both to their location in the cell, but also because they have slightly different biophysical properties. So what I want to talk about today is two main topics that we've been looking at recently. One is our recent work looking at melanoma, and we've more recently branched out into looking at other kinds of cancer, but I'm going to focus on melanoma today. And then later on, I'll talk about our work looking at the role of TPCs in sperm. So our interest in melanoma mainly came out through a collaboration with Antonio Filippini, who was at the University of Rome, and he's had a long-standing interest in melanoma, but we were interested to see if there was a role for TPCs in melanoma tumorigenesis and metastasis. And obviously, as a clinical problem, melanoma is a big problem. There's a rise in rates of melanoma. This is despite the fact that obviously one of the key risk factors of melanoma is exposure to the sun. But unfortunately, despite that, there are rising levels of melanoma in the population, probably linked to the way that, for instance, people in Britain and other countries in Northern Europe started to go on holiday to places like Spain and got exposed to the sun. In general, not really, obviously, protect themselves, but it's a problem, and it can also be a major problem in terms of melanoma then developing into a malignant form. So this is just showing the different stages of melanoma, initially confined to a specific region of the skin, and if it's obviously caught early enough, surgically, it can be possible to remove that and there's no more problems. But if the tumor has started to spread, and particularly to metastasis, then obviously that's a major problem. So there's lots of interest in trying to understand the biology of melanoma, tumorigenesis, but also metastasis. And there's still lots of unknowns about the metastatic process for cancer in general. So you could argue that this work is also relevant to cancer in general. 90% of all cancer deaths are due to cancer cells spreading around the body through metastasis, and yet we still only incompletely understand what metastasis involves at the molecular level. So this is some of the mechanisms of metastasis. You can see there's different stages. First of all, you have motility and invasion. Then there's an interesting phenomenon called plasticity, where the tumor then starts to adapt itself, as it were, to its changes in circumstance. It then starts to modulate its microenvironment, and there's actually some very interesting evidence, I'm sure you'll be aware of, that tumor cells can start to reprogram tissues around them using EVs, and that seems to be one way in which they modulate the cells around them, and that allows them then to colonize tissues and start to then spread across the body. So lots of interest in understanding what we mean by metastasis as a way to try and develop new drugs to treat cancer. Now, in terms of melanoma, there's already lots known in terms of the molecular basis of melanoma. In particular, MITF, a transcription factor, plays an important role, and there's also a role for other signaling proteins. This is the MAP kinase pathway, and in particular, mutated forms of RAS seem to be linked to specific types of melanoma. And there's also a model that I'll return to much later on in the talk called the rheostat model, which is basically saying that the levels of MITF, this transcription factor, have implications in terms of the state of the cells. So depending on the level of MITF, you can either have a senescent cell or undifferentiated cell, but you can also have invasive and proliferative cells as well. And so this has an implication, obviously, in terms of the ways in which melanoma cells start to become tumorigenic, because MITF plays a particularly important role in melanocytes, which are the cells from which melanoma cells are derived. So we decided to start looking at this problem because we have this interesting two-part analysis, primarily by knocking out TPCs, and then to see what then that effect that has on the melanoma cells properties, characteristics. And this is work that we started with Antonio Filippini, as I said, this first part of the work. And so what we did was initially we used CRISPR-Cas genome editing to knock out TPC2, and then we screened for cells that had the knocked out gene. We analysed this by sequencing and mutational methods. And we showed that we then got mutated TPC2 knockout melanoma cells. So what we're doing in this case is looking at a human melanoma cell line called CHL1. I'll come back to talk a bit later on about another cell type called B16, but this is CHL1 human tumor cells, melanoma cells. And what we've shown here is that some of these cells have got the knocked out TPC2 gene. So what we did first of all then was to look at the effects then of TPC2 knockout on different properties of the melanoma cells. And what we showed, one of the first things we showed was that this was actually quite surprising, because up till this point, based on some other work that people have done on TPCs, and the effect of knocking out TPC2 on cancer cells, it was mainly assumed that if you knock out TPC2, you're likely to have a beneficial effect. You make the cell less tumorigenic, less metastatic. And so it was quite a surprise then that actually TPC2 knockout seems to make things worse. So in this case, it's impairing adhesion, the extracellular matrix. As you can see here, what we're looking at here is the adhesion of these cells to the extracellular matrix, and we're also looking at the link with integrin, which is part of this process. And so the knockout is actually making things worse. So that was quite a surprise. And in line with that, we showed that TPC2 knockout in these CHL1 cells increases metastatic cell invasion. So this is using an in vitro assay to measure this invasiveness. Now, that then led us to think, well, what is the cause of this increased metastatic potential? And we decided to go looking for links with a cell signaling pathway called the Hippo pathway, mainly because this has been shown in recent years to be a key regulator of malignancy. So the Hippo pathway is an interesting pathway. It was originally discovered in Drosophila and plays an important role in the formation and the size development of organs. But it's also been shown to play all sorts of important roles in tumors. Now, one of the interesting things about the YAP-TAZ, the Hippo pathway, and the downstream activators of the Hippo pathway are YAP and TAZ, is that they have quite a dual role. So in some circumstances, they can play an oncogenic role, in others, a more tumor suppressive role. So we decided to go looking to see if we could find a link here with the Hippo pathway. And we found that indeed YAP and TAZ are so this I should say, by the way, I'm skipping ahead here. So this is the Hippo pathway, key regulator malignancy. And essentially what happens is when the Hippo pathway is active, then YAP and TAZ, which are these transcriptional activators that are linked to tumorigenesis and metastasis, are active. And when the Hippo pathway is suppressed, then these become active. So that's been known about. So what we then looked at was the effects of TPC knockout then on the YAP and TAZ pathway. Actually, what I'm showing here is TPC2 knockdown. So we've shown, and this is all published by the way, we've shown that TPC2 knockout will activate the downstream effectors of the Hippo pathway. So these are genes that are induced by the transcription activators YAP and TAZ. And what we've shown with the knockout is that we activate these genes. And in fact, we've also shown a similar thing with TPC2 knockdown using siRNAs. So this is a good demonstration that by two completely independent methods, knocking out or knocking down TPC2 will activate the YAP and TAZ pathway. So the question then is, well, what is a calcium channel, which is on the lysosome, have to do with this pathway? And this is the bit where we're still, in a way, trying to work out what's going on, because this is the Hippo pathway. As I've said, the Hippo pathway is involved in suppressing YAP and TAZ activity. And there are some interesting recent findings showing that there may be a link with calcium signaling, in that the ORI pathway has an activated role in the store operative calcium entry system, which is regulated by ORI and STIM1, which is essentially when calcium comes into the ER from this outside the cell in response to too low levels of calcium in the ER. And ORI and STIM1 are regulators of this process. And there was some previous work showing that this pathway, this store-operative calcium entry pathway could activate the Hippo pathway. So we thought, well, given that our channel is a calcium channel, could it be there's some kind of link there with this process? Because this was at the time the only kind of non-calcium link we could find. And indeed, what we found was that we do have downregulation of ORI1 in the CHL1 TPC2 knockout cells. So at the moment, what we think is happening is that when you knock out TPC2, that then for somehow or other then leads to a downregulation of ORI1, which is store-operative calcium entry. This stops calcium coming into the cell, and in consequence, the Hippo pathway is deactivated, and this then leads to the aphantase proteins being activated. But at the moment, we're still very unclear about how exactly a loss of an endolysosomal calcium channel is leading to this change. Now, the next bit of the talk I want to talk about is what happened when we started to look at TPC1. So as I mentioned earlier on, TPC1 and TPC2 differ in both in their location within the endolysosomal compartment, but also in their biophysical properties. So this is unpublished work, and at the moment we are trying to get this submitted for publication, and it's basically looking at what happens when we knocked out TPC1, not TPC2 in this case, but we have done some TPC2 work as well in this study. What happens when you knock out this gene in a different melanoma cell? In this case, it's a B16 mouse melanoma cell. So what we found was that if you knock out TPC1, you have some kind of interesting effects on the cell. So on the one hand, we boost the production of melanin, so that was kind of interesting. But this is the effect we have when we knock out TPC1. And as you can see here, this is due to, so there's a lot of information here, so I'll try and go through this as much as I can. When you knock out TPC1, you boost melanin production, and this is due to production of overexpression of enzymes that are then involved in melanin production. Now, this is interesting because in general, if you boost melanin production, this is the sign that you are having this effect on a differentiated state of the cell. And then the other thing that we found that was interesting was that when we then start to look at proliferation, we found that we also have an effect of TPC1 knockout on this process as well. Unlike the situation with TPC2, where we don't see this in these cells. So what are we talking about? So sorry, we don't affect proliferation, but what we do affect is this melanin production here. So this seems to be one thing that would fit with the idea that TPC1 knockout is having an effect on the differentiator of the cell. So then we started to look at the effect on the proliferation of these cells. Okay, so this is important because obviously what we've shown in the past study was that TPC2 knockout has an increase on the metastatic potential, kind of invasibility of the cells, that kind of thing. The difference with TPC1 seems to be that if you knock out TPC1, you have the opposite effect. You decrease the proliferation, the migration, the invasiveness of the cells, as you can see here, because what we're basically showing here is that unlike the situation with the wild type and the TPC2 knockout, when you knock out TPC1, you decrease the proliferation potential, but you also have an effect on the proliferation of the cells, so on the invasiveness of the cells. So the cells are less able to invade. For instance, this is a wound healing assay, less able to invade, say, a wound, and also with the measures as well that we've done of invasiveness. So go along with the idea that what we're doing here is decreasing the invasiveness of these cells. So why is this? Well, that got us thinking then about what's known about proliferation, metastasis in melanoma cells. And we decided to look at MITF, which I've already mentioned is a key player in melanoma tumorigenesis. And also another factor called TFEB, which is a related transcription factor. And what we found in this analysis is that MITF, particularly an isoform that's only found in the melanoma cells, MITFM, are upregulated with TPC2 knockout, whereas the effect of TPC2 knockout is not any different to wild type. And we've also shown that TFEB, which is this related factor, is also upregulated, is also more active in these cells. So that could explain why we've seen this interesting effect on the melanoma cells, because as I said earlier on, according to the rheostat model, MITF, if you overexpress it, can actually suppress, can promote differentiation and suppress tumorigenesis, which could explain why we see an increase in melanin production, as well as decreased proliferation. And that then led us to think, well, why is that? What's happening here? And what's it got to do with TPC1 knockout? Now, what we decided to then do was to look at factors that might be somehow responsible for this. And what we found that's interesting is that we have a factor called mTORC1, which is known to have an impact on TFEB and MITF. Basically, when you have high levels of mTORC, then it suppresses TFEB and MITF. And what we've found is that we've got a downregulated mTORC1 in these cells, as shown here. I think shown by two different things. One is that the downstream proteins are activated by mTORC1 are affected by TPC1 knockout in a negative way, but not by TPC2 knockout. And also we looked at a thing called Raptor, which is a protein that's associated with mTORC1, and this is all missing in the TPC1 knockout cells. So this is all in line with the idea that mTORC1 is basically inactive in these TPC1 knockout cells. And so at the moment, we're then thinking this as a model of what's happening here, is that in the normal situation, mTORC1 somehow associates with TPC1, and there's actually some evidence that mTOR can interact with TPC proteins on the lysosome. So normally, you have a situation where the mTOR1 is active, and that then suppresses MITF. But then in the knockout situation, we've got a situation where the mTORC1 is downregulated, and this allows the MITF and TFEB to then work in the way that they do. Now, what we still don't really understand is exactly how this all fits together. I mean, one of the problems, obviously, with these studies is that what we do here is we're knocking out the gene, we then see what happens in a cell, and we try and then, on that basis, to come to some conclusions about the normal role of that gene within the cell. But what we still lack is an idea of what else might be happening in the cell. So we started to do other things like overexpress TPC1 and TPC2 as another way of looking at this situation. And ultimately, I think we need to explain, how it is that an endolysosomal calcium channel is mediating these effects. Is it linked to calcium signals and that kind of thing? And that's something we're still very much unclear about. Other work that I'm not going to talk about today is starting to look at what happens when you inject these cells into living mice, because obviously you can learn quite a lot from in vitro assays in cultured cells, but ultimately we want to know more about it in the actual living animal. And we've been injecting these wild type and TPC2 knockout and TPC1 knockout melanoma cells into mice to see what happens. And at the moment, our preliminary evidence is that instantly the wild type tumor grows a lot bigger than the TPC2 knockout tumor in a living mouse, which was interesting because we didn't know what to expect based on the in vitro work. But surprisingly, despite the TPC2 knockout tumors being a lot smaller, they're much more toxic to the animal. And we don't know why that is, and we think it could be some inflammatory response, or maybe it's something that's coming from the tumor cells and moving around the body. So obviously EVs would be something that we're quite keen to look at in that context. Right, so that's the work we've been doing on the cancer. Now, what I want to do for the final part of the talk, the second part of the talk, is to completely switch to a different topic, but it's obviously one pertinent to some of the work that's going on in the department and the defence of the thesis we're going to hear about later on today, which is to look at sperm biology and fertilisation potential. And this is a subject that I have had an interest in for many years. I mentioned that I had a big interest initially in egg activation. And so it was fairly natural then, having made, say, TPC2 knockout and TPC1 knockout mice, to then see if we had any impact of TPC2 knockout and TPC1 knockout on the sperm. And we're starting to find some quite interesting things there. So this is just a bit of background about some of the processes that involved at fertilization. A key thing that we're going to hear more about later today, actually, is that sperm are not initially ready to fertilize the egg. If you take an ejaculated sperm and add it to an egg, it will not fertilize the egg. What has to happen is that you have this process of capacity, which normally occurs in the female reproductive tract, but can be triggered to some extent artificially in an ART clinic. And this, it's only after this that the sperm then becomes capable of fertilizing the egg. And there's a number of different things involved in capacitation. One is that the sperm become more motile, hyperactive, and they change in motility. And they also then become capable of fertilizing the egg by being able to get through the zona pellucida and also to bind to the egg membrane after undergoing the autosomal reaction. So we wanted to look at then the effect of knocking out TPC2 on sperm biology. And so one of the things that really caught our interest that led us to work on this was a student of mine called Alapak Chen, who also was one of the ones who worked on the PLCZ, but he was the one in my lab who made a PLCZ to knockout mouse and showed this was incapable of inducing the sperm, incapable of inducing the calcium oscillation and fertilization. But he also then started to look at TPC to knockout sperm, and he showed that the TPC to knockout sperm had interesting changes in their motility. In particular, they had differences in motility. So this is the red is the knockout sperm, and the what blue is the wild type sperm. He showed they had interesting change in their motility that seemed to be more like a hypomotile state, a bit like you might normally find with capacitation. So that got us interested in why this was. Why is it that knocking out a gene has this impact on sperm to make them more motile than normal? Because I think we thought, I don't know if we were so naive about this, but in general, you'd think when you knock out a gene, it tends to have a detrimental effect on the process. So we found it interesting that in this case, it seemed that the sperm were actually faster than normal, as it were. And we then started to look at why that was. So one of the things that we thought we'd look at was whether there's any difference in energy production between the sperm. And in line with this, what we found when we did an ATP analysis assay was that indeed the TPC knockout sperm have got substantially high levels of ATP. Obviously, this is the energy currency of the cell, so it's interesting that they have these dramatically increased ATP levels. And then to look at why that might be, we use analysis using a seahorse analysis system. And we found that the TPC knockout sperm have apparently increased oxidative phosphorylation. So this is looking at a measure that looks at glycolysis, this is looking at a measure that looks at oxidative phosphorylation. So it seems as if something's happening in the mitochondria to boost the production of ATP via oxidative phosphorylation. Now, looking at what might be the cause of this, we started to look at whether there was some difference in the energy, the substrate utilization of sperm. So this is work done by my student for Cameron. And what he did was to look at a number of different substrates and see if they had any increased utilization in the knockout sperm. And what he found was that there's a substrate called G3P, which is increased in sperm. And so this may be one of the reasons what we're seeing here is happening. And in fact, there's an enzyme that normally G3PDH, which is an enzyme in the mitochondria, there's an enzyme that utilizes the substrate, G3P, which seems to have higher activity in its sperm. Now, this is interesting because there's some evidence that this substrate can funnel, I mean, it can play all sorts of roles. It plays an important role in gluconeogenesis, but it also plays an important role in oxidative phosphorylation. It's one of the ways in which you can boost oxidative phosphorylation. So this could be one of the reasons why we're seeing this increase oxidative phosphorylation in the sperm. Now, the other interesting thing that we found was that, well, what we expected was if you've got this increased oxygen utilization by the sperm, if you've got this increased oxidative phosphorylation, increased mitochondrial activity, could this be detrimental to the sperm? You might think there'd be an increase in reactive oxygen species, for instance, which obviously can be quite detrimental to sperm. And yet we were surprised when we did analysis of these reactive oxygen species that we didn't see any increase in the knockout sperm. If anything, we seem to see so this is a treatment that will induce this. reactive oxygen species, and what we found actually is that they are, sorry, this is when you get, I say sperm. So it seems as if the TPC knockout sperm actually got lower level of energy than reactive oxygen species. Now, the reason we think this may be is that when we then start to look at enzymes that are involved in suppressing reactive oxygen species and protecting cells from oxidative stress, that's these enzymes, SOD1 and SOD2. These are dramatically increased in expression in the knockout sperm. So we think this is one reason why the RS levels are not high as you'd expect. And also we think it could be interesting from an opposite point of view from the egg, because there is some evidence that during the process of aging in women, when egg quality starts to decrease as women age, one of the things that's been linked to this is high levels of reactive oxygen species in the aged egg. So it was interesting talking to a colleague at a conference at the Gordon conference earlier in the summer actually, who thought that it's quite possible that maybe we'd see something similar going on in the eggs of these mice, and we thought that would be interesting to look at, because if that were the case, it might mean that the aged female of this knockout of these knocked out in this TPC knockout females might have then lower levels of RS in the eggs, and that might actually then lead to them being have protective mechanisms for aging, which obviously could have implications in terms of therapy and things. So that's something we thought would be quite nice to look at. The other thing we thought we'd look at then, because we found that the TPC knockout sperm have enhanced motility, all the things that you tend to associate with capacitation, was we decided to look at then whether there was any evidence that the TPC knockout sperm have enhanced parameters that we would normally link to capacitation. And in fact, what we've found is that things that are normally only found in the capacitated state, which is, you know, that high levels of pKa, the enzyme pKa, and also phosphatyrosine, tyrosine phosphorylation of proteins in the sperm. These are things that are increased in the knockout sperm. And so this is in line with the idea that what seems to be going on here is that things that normally only happen in capacitation also seem to be happening in the knockout sperm. So this is showing the tyrosine phosphorylation. That was the pKa activity. So what he's showing here is that the tyrosine phosphorylation is increased in the knockout sperm. And in line with that, we've also found that the uncapacitated TPC knockout sperm enhanced fertilization rates. So what we're looking at here is a positive control in which we've got wild type sperm that are being capacitated. We then got the uncapacitated wild type sperm. You can see there's some fertility. Maybe this is due to spontaneous capacitation, but obviously you normally expect uncapacitated sperm not to be able to fertilize the egg. But what we found was that the TPC knockout sperm, even in the incapacitated state, we see much higher levels of ability to fertilize the egg. So it seems as if somehow when we knock out the sperm, not only do we increase the energy levels and the motility of the sperm, but we're also, the sperm seem to be behaving more like the capacitated state. And we, at the moment, we want to try and understand what that's got to do with TPC knocking out a lysosomal calcium channel, particularly obviously in the sperm. You don't really have a lysosome as such. You have other kinds of sperm specific organelles. And at the moment, we've got this hypothesis that maybe cat sperm, which is a protein, a calcium channel on the sperm surface that's involved in capacitation, may be more active in the TPC knockout sperm, and that may have some link there with the fact that calcium signaling in the sperm has changed. So lots of things still to look at in terms of understanding what's going on in the sperm. So I just thought I'd summarize then everything I've talked about so far, and then it'd be interesting to hear people's views on what some of the things we've been seeing. So I think some of the first things to say is that TPC1 and TPC2 play quite differential roles in cancer progression. I mean, there's a body of work now looking at the effects of knocking out TPC1 and TPC2. Actually, very little work on TPC1, but lots of studies looking at the effects of knocking out TPC2. And certainly in some circumstances, knocking out TPC2 seems to have a beneficial effect in suppressing tumorigenesis and metastasis. But what's clearly happening in the CHL1 cells that we're looking at is the opposite, that we see an enhanced metastatic potential when we knock out TPC2. And we think this may be partly related to the stage of tumorigenesis that these cell lines were derived from. So we think it's interesting, for instance, that the B16 cell line, for instance, is a cell line that comes from an early tumor, whereas the CHL1 cell comes from a more mature metastatic tumor. So that could be one difference. But we also think there's some interesting links between other factors in the cell, the background levels of MITF and those kind of things. And as I said, I'm not showing the work today, but we've also got some interesting, slightly confusing and contradictory findings when we inject these cells into mice. On the one hand, we have TPC2 knockout cells that are less capable of forming a big tumor. They seem to lead to a reduced size of tumor. But on the other hand, it's much more toxic to the animal. So why is that? And I think what all this shows is that TPCs may be important new drug targets for cancer treatment. And there's already been work trying to identify inhibitors and activators of TPC2, much less so TPC1. We've got an interesting TPC1 for all sorts of other reasons that I've not talked about today. So we're also trying to make pharmacological agents that will inhibit TPC1. And what these might have potential for in the future is as therapeutics. But as I've already said, that the context in which you knock out TPC2 seems to affect what happens to the cell. We'd have to be quite careful if we're going to use these as therapeutics for cancer treatment, that we get it right. You don't want to add an inhibitor to give a drug that inhibits TPC2 and then find it makes things worse in terms of the cancer. Moving on to what I've talked about in terms of the function of TPC2 in sperm, there's this rather surprising and unexpected initial finding when we found that this actually increased motility and energy production. So again, we still want to know why that is. And it's interesting, actually, this isn't just something we found in sperm. We've started to look at other tissues, other cell types and other tissues, organs from TPC2 mice. And we also see this boost in TPC2 levels. We've also, linking what I said earlier on about mTORC1, we've also started to find some interesting effects of knocking out TPC2 on markers of longevity. And this is interesting because if you knock out mTORC1 activity, there's an interesting finding in the past that's shown that this seems to boost longevity in certain contexts. So I think it was, the worms where they knocked out mTORC1 and they showed this boosted the lifespan of the animals. And so there's a whole kind of interest emerging really about the ways in which things like mTORC are linked to nutrition sensing, but also longevity and aging and sort of things that we're getting quite interesting in our lab really. And I think this all shows that, the idea that people used to originally have of the lysosome as just being this, dustbin of the cell, this trash can of the cell, it's really quite a simplistic idea that the trafficking system in the cell it plays all sorts of important roles in transporting substances around the cell, but also it's there to sense the nutritive state of the body of the cell. And maybe there's links there with aging and longevity, which could be quite interesting. So how we kind of tease all this apart in terms of understanding how TPCs work is we're still very much at the start of all this. But what we're starting to understand is that endolysosomes are very complex organelles. And I think the fact there's an interest in this department in EVs is interesting in that context, because of course EVs are linked to the endolysosome trafficking system. So I think in the future, we'd have a big interest really in trying to understand how exactly the cell signaling aspect of TPCs fits with their role as proteins located in endolysosomes, and how that might then fit in terms of the transport of substances, not just within the cell, but between cells. So I'll leave it there. I hope that makes sense. But I'm not, and it kind of, because it's obviously quite a number of different kind of elements there. These are quite new angles for our lab, so we're still very much trying to feel our way into what all of this means. But anyway, I'll leave it there. Thank you. Well, I should thank some of my collaborators. So I should end by this, which is that these are people who are involved. Ali and Bash is a student at the lab who did a lot of the cancer work with Antonella Del More from Antonio Filippini's lab, and Al Chen did the work on the sperm, then Shuhui Jin's doing that. Colin Gordon's work with us on the melanoma work, and these collaborators have helped with the aspects of the sperm cancer work. Thank you.
Thank you very much for the interesting talk. Now the floor is open for questions. Yes.
Thank you very much for the TCP2, especially the part that you were speaking about the sperm and how it is someone making the sperm to go actually a super sperm to was you have knocked it out. And is there any possibility to use inhibitors or chemicals or something? Because if that would be, can be maybe a medicine that can be used for the treating of the people that have infertility as a result of the male.
Actually, I should have mentioned that actually my student has shown similar things with using inhibitors. Yeah. Actually, he's defending his thesis on Tuesday. Actually, so he's going to talk about this a lot in his viva. But yeah, so that is interesting. That actually we can more or less mimic the knockout situation with inhibitors of TPC2. Yeah. So maybe it would have therapeutic potential in that sense if you could boost,
that inhibitors. I don't know, are they toxic?
I think the problem with inhibitors, and this is relevant to the cancer work, is they're not that specific. So there's a real interest in trying to develop much more specific inhibitors of TPC2 that are don't have some of the effects on other signaling pathways that they used to. Actually, my wife is now working almost primarily on Alzheimer's and Parkinson's, so neurodegenerative diseases, but because she used to work on this area with myself and my collaborator Anthony Alione, she's at the moment doing a large-scale screen trying to identify much better inhibitors of TPC2 and activators as well. So while they've got a particular interest in the link with Alzheimer's and to use these therapeutically in that case, obviously they would have all sorts of interesting potential uses as well if she can come out with something a lot more specific.
Thank you for a very interesting talk. Perhaps a simplistic question, but from just simple genomics, what is there on the TPC2 channel with regard to melanoma? Is that known, for example, like different SNPs or whatever that change their levels?
Yeah, we looked at that and we found that we published a study actually of looking at polymorphisms in TPC2, and we showed that in the UK Biobank, and we showed that there are polymorphisms in TPC2 that make you more likely to have some kinds of cancer, and other polymorphisms that make you more protected against cancer. I'm trying to think now with the melanoma. I don't think there was anything that really stood out though as it being like, wow, this is a sort of super link. I mean, there's definitely a link emerging with TPC2 and cancers, all sorts of different kinds of cancer. But I probably need to go back to that paper and check it out.
From your in vivo work, right? To try and
now the in vivo work was interesting because I guess what we expected was we basically knocked out TPC2 in these B16 cells, and that has an effect of not leading, so that basically suppressing the metastatic potential of the cells. And then we knock it out in CXO1. Sorry, you were going to say?
But it increased melanin and increased tyrosinase activity. Did I remember that correctly?
No, that was a TPC1. Yeah. So the TPC, so in B16 cells, TPC2 knockout is actually it's a quite small effect, but if anything, it sort of makes them less likely to metastatic. Whereas in CXO1 cells, it has a big kind of effect of making them much more aggressive and metastatic. So we kind of expected that if we injected B16 cells or CXO1 TPC2 knockout cells or wild type cells into mice, we might see something that was more showed that in vitro difference. But what we actually found was that the B16 knockout cells and the CXO1 knockout cells, obviously we're injecting the human cells into new mice, so it's a bit different. But in both cases, it's pretty similar effect. What happens is that the wild type tumor grows much bigger. The knockout tumor growth is much smaller. It's almost like it gets to a point where it just stops growing, and that in itself could be linked to kind of effects of the on proliferation of spores. But what was interesting then was to find that those smaller tumors are far more toxic to the animal. So the organs start to show sorts of big signs of damage in the liver and in fact, spleen, everything really. And that was a bit of a surprise thing because we thought, knockout tumour being smaller would be sort of less toxic. So what we don't understand is why that was happening there. And also why it seems to be the same for the B16 or the CF1 stance. But I think it's made us aware that having those in vivo analysis as well as the in vitro analysis is pretty important, really, because you can assume all sorts of these image classes.
Any more questions? Do you think that maybe this might have some kind of immunomodulatory functions, that why it's more toxic? Do another immune response to.
Yeah, possibly. And in fact, we have looked at cytokines and found some differences there that might fit with that. Yeah. I think what we still haven't quite got is that kind of clear mechanistic link, but that's what we're starting to look at now. Yeah, so it's quite possible it's an inflammatory response. Yeah. And we also thought because endolysosomes and EVs, there's obviously an interesting link there. It might be those tumours are sending out EVs that are having some of these effects.
Like we should be there, we have questions from online participants.
Okay, yeah.
So I see two chats.
That was earlier.
Sorry. There's no questions. I would like to invite Professor Andreas to hand over the certificate to.
I'm making a picture too, and this will be your hands. Thank you very much. So thank you.
Thank you very much. Thank you very much for the participation. Have a good day.
I would like to, yeah, anyone that wants to come to